I'm not going to go into this in detail because there's no need to, but I just wanted to touch on some words. Affinity is the main one, and specificity. So affinity, how strong the interaction is for the target between the ligand and the receptor. And by receptor, I just mean the thing the ligand is binding to. Okay, big, normally small thing and big thing. I also stole these slides of someone else, and I don't agree with all of them. KD does not tend to be micromolar for biological systems. That's nonsense. Sometimes it is, sometimes it's nanomolar. Anyway, also for a drug, we don't necessarily want to be as strong as possible. So we're going to argue with that. I'm not going to discuss it. One of the important bits as a biophysicist, KD and KI, if you can make them the assay independent versions rather than the IC50, that saves a lot of problems later on. Specificity, binding to the target as opposed to related targets, can give you side effects, but can also be polypharmacology. Sometimes you get a better effect if you hit multiple targets. Dirty kinase inhibitors can work quite nicely. So that's something to think about. One of the really interesting ones, though, is mode of action, because over the last 5 years or so, we've now gone from the idea of kind of an antagonist or an agonist, an inhibitor and an activator of some sort, through to chemically induced proximity and bifunctional molecules that can bring two proteins together normally, or bring two components in the cell together. So chemically induced proximity is a really interesting mode of action, where what you're looking for is almost now just a warhead that binds, a true ligand. And then you can recruit cellular factors to have effects on your target. So for example, you can recruit a kinase or a phosphatase to change the phosphorylation state of your protein. That's been done quite nicely. The original versions are all PROTACs, which is where you're recruiting E3 ligases to ubiquitinate your protein and get it degraded by the cell. And then that loops back round to thalidomide and phase 4, which is where that all came from, and that understanding of the degradation pathways. So, but also, what we want is a drug. We don't just want a ligand. We don't just want to bind to the protein or the target. We want to have an effect in a biological context. So we need to move beyond a ligand to bioavailability, to considering how that small molecule, which is what I'm just going to focus on, how the small molecule is behaving in the biological system. So it's behaving normally in the human body. And in particular, the PK/PD, pharmacokinetics and pharmacodynamics, what the body does to the drug and what the drug does to the body. And those kind of interactions is where we move from that kind of really being just a ligand through to an actual drug. It's that balance of activity. Resistance mechanisms, it's not bother, we've lost a few minutes. And tolerability, everything's toxic. It's fine, life's like that. So, target discovery. You've got to decide if you're doing a target-based drug discovery program. You've got to think about at the beginning what are you trying to treat? How is it going to be? The brutal part of that is, well, so there's acute versus chronic. Does these manifest? What are the existing options for treatment? Are we going for a best-in-class, first-in-class drug, or how are we going to follow on? But also, what does society need? What will it tolerate? What will it pay? You know, at the bottom line for an awful lot of commercial drug discovery, you're coming back to what would it pay? And that's, you know, just I wanted to touch on this because I think it's interesting and people gloss over it sometimes. But, you know, there's been an awful lot of discussion of obesity diseases. That's very definitely a disease of the first world. Tom Lehrer, you may remember, specializing in diseases of the rich is a line which keeps floating up in my mind every now and then. So you need to focus on a disease. There is a lot of commercial incentive to specialize in diseases of the rich. But just to go back round, one of the most important drugs in the last century is just oral rehydration therapy. It's a drug. It's just, it's a drug in that has a physiological effect on the body. If you've got diarrhea, that is the best thing by far. 50 million lives, 50 cents a course. It's a very effective drug. So it doesn't have to be a state-of-the-art small molecule induced proximity thing. Can be, and they're quite cool. And there are some diseases which definitely need that. But there are some where it's just salt or diet. Anyway, that was a slight digression. Target discovery. So you need to have found your target. So normally from looking at disease states, from looking at often what proteins or what cellular targets are upregulated, downregulated, mutated, modified in the disease state. A lot of that's tied through to genomics at the population level, or at the individual level, or at the tissue level. There's been some really nice cancer genetics looking at the variability of the cancer genome across a tumor, and the way that you get that really rapid divergence of the progenitors turning into just a wide variety of mutations. Target validation, CRISPR, inducible knockouts and knock-ins to try and find out if the target is worth studying, are incredibly useful. A lot of what we do in the kind of the biotech area, especially collaborating with people, is generating proof of concept compounds. So not drugs, not all the way through to phase 2, but just getting a compound which has cellular activity, which you can then say, does this modulate the target the way I want it to? Is this target validated and worthwhile pursuing all the way through to make a final therapeutic? And it's hard to nail down where that is, but essentially it's that kind of advanced lead stage is really key for deciding whether or not the target is going to validate, and it's whether it's worth investing the remaining 95% of the money that it's going to take to get it through clinical trials. So I said I was going to come back to phenotypic screening because it is, a lot of drugs came from phenotypic screening. It's actually really interesting. It's also quite a nice way of doing target ID and target validation. So if you have a clearly defined phenotype, and that's the key to phenotypic screening, you can start to screen for compounds that modulate that phenotype. Dead is a really bad phenotype. So we do anti-infectives quite a lot, and people say, well, just look for things that kill bacteria. There are lots of things that kill bacteria. Bleach kills bacteria. That's no good as a drug. Boiling kills bacteria. It's no good as a therapeutic treatment. So there are lots of causes of dead. Lots of causes of dead, not of drug. But there are lots of causes of dead. Dead is a bad phenotype. But if you've got a good phenotype and a good phenotypic assay, you can definitely drive it through and drive that drug development cycle by improving the compound, making it have a bigger effect in the assay. The problem is that if the mode of action changes and you start to have an effect which looks like the one you were having, you don't necessarily know. And there's some really nice examples of people who have just ended up chasing what looks like a very promising phenotypic hit, and then you just end up chasing it and it turns into grease, because grease is always active in phenotypic assays. But again, grease is not a drug. So understanding the mode of action and being able to dial down into the mode of action is really, 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 really important. So knowing how your compound is having the effect either on the target or on the biological system. So once you've found a compound that has that effect on the phenotype, that could be quite a nice way of then saying, okay, what's the molecular target? So we could then take a target-based approach and say, okay, we've got a compound which affects the phenotype, but it's not ever going to be a drug, and it's going to be hard to turn it into a drug, but we can use that compound to pull out the molecular target that can give us the correct phenotype, which is going to make it easier to turn that into a drug. So I've got two examples of that, fumagillin and ribocil, which are two kind of nice phenotypic evolved hits. So fumagillin is that compound at the top, which is horrific. It was discovered in 1949. It's a bacterial product, a natural product, and it's an antimicrobial. It's been touted as for inhibiting malaria, but at some point, and in that kind of classic style of people throwing things on the cells and see what they do, or throwing them at organisms and seeing what they do, they found that that compound inhibited angiogenesis, which is heavily involved in tumors, especially solid tumors. So That compound inhibited angiogenesis, as well as being an antimicrobial. It had this other slightly unexpected effect. So, group at Yale made a labeled version of that by coupling a biotin onto the acid. Worked with lysates, so made a column with it, made some lysates, put that of cell types that it was active on, blow them over the column, see what it pulls out, fish it out, do some proteomics, find out what the compound is. They identified methionine aminopeptidase as the compound that, as the protein that that compound inhibited in tumor cells, and then that's now been quite a successful oncology target in its own right. So that's quite a nice way of going. We've got a phenotypic hit. That thing at the top is in terms of making that on a large scale or doing some SAR around that, it's going to be awful. But we can use that as a tool to find the correct compound. The second example is ribosyl. So I started off doing my drug discovery against RNA, not proteins. So that's every now and then I may say, typically proteins. We started off doing small molecule RNA, but at the time it was kind of unvalidated. 2015, this paper came out. I think this is, for me, this is a beautiful example of phenotypic. So a group at Merck were looking for anti-infectives using phenotypic screening. They were focused on the riboflavin biosynthetic pathway. So specifically the riboflavin biosynthesis in E. coli, because what they'd observed in their labs was that coli in an infection setting were unable to scavenge enough riboflavin and they died. So if you had a strain which was deficient in producing riboflavin, it couldn't survive in an infection environment. It was fine if you slosh it around in a 10 ml flask, but if you stick it in an infection, it died. So what they thought was, okay, if we can inhibit the riboflavin biosynthesis, that could potentially be a really nice antibacterial. Okay, so they did a quite small screen, 57,000 small molecules. But the nice phenotypic screen they did, so I said dead is not a good phenotype. Dead was one of their phenotypes. But what they were looking for was a recovery where you add riboflavin to the media and then you get rid of that antibacterial activity. So it kills bacteria, but if you supplement it with riboflavin, that antibacterial effect goes away. Okay, so you can actually say, like, this is specific in some way to the riboflavin biosynthetic pathway. But you're not limiting yourself in saying, okay, it's got to be this enzyme or that enzyme, because there's always turns out to be redundancies. So what they did, the compound is ribosyl, which is this thing here. When they dialed down and found out what it bound to, it binds to RNA, not to a protein at all. So the gene, the mRNA controlling the riboflavin biosynthesis is controlled by a riboswitch, which is that thing there. Riboflavin binds to it, folds the riboswitch, and prevents the mRNA being translated. This compound binds very effectively to the riboswitch. So the bacteria go, we don't need to make riboflavin. The switch is occupied. They never make riboflavin. They die. So really, really, really elegant identification of a target that they never would have thought was a viable target, but also a whole mode of action which was kind of potentially really interesting. Unfortunately, it's absolutely no use as an antibacterial, because as soon as you put any evolutionary pressure on it, resistance pressure, they just change the base usage in the riboswitch, and they just evolve out of it. But the paper goes on to say very nicely that if you have a therapeutic area where you don't have a strong selection pressure, so almost anything other than oncology or anti-infectives, that's a viable target. The riboswitches are potentially a viable target. And in fact, RNA now, again, in the last 5 years, has become a really interesting and hot area for small molecule drug discovery. Yeah, so identified RNA binding as a viable mode of action. Right. I'm going to plow on because I'm having spent 10 minutes randomly talking at the beginning with no slides. I'll focus a bit more. Hit ID. So you've identified your target. You need some chemical matter that you're going to try and evolve into a drug. So there's different ways of finding your chemical matter, literature inspired. is a simple way. If someone like the fumagillin example, if you know from the literature that there's a compound that binds to your protein or your target, just use it as a start point. There's absolutely nothing wrong with that. Mechanistic transition state inhibitors, some of the best enzyme inhibitors are transition state inhibitors. It's old technology, but it's incredibly powerful. High throughput screening, got a good assay, you screen 1 million compounds, 5 million compounds, whatever, look for inhibition of that. Fragment-based screening is what I've been doing for the last 25 years. It's fragment-based approaches. Very elegant, has some limitations, has some great strong points. Different way of doing it. DNA encoded libraries, what I've been doing for the last 2 or 3 years. Also very interesting. Again, has some drawbacks. There are no panaceas, but it's a very nice way of finding new chemical matter. ASMS, affinity in solution mass spec, very nice way of screening in an unbiased way for interactions between pools of compounds in particular and any molecular target. So a lot of people use ASMS to look for RNA binders, for instance. Virtual screening, and especially AI virtual screening, also incredibly popular, and really a lot of money is being thrown at AI to try and generate hit matter, because the rest of the ways A don't work and B are very expensive. So the key bit, one of the key bits, the first key bit before we go on to the next key bit, is that you've got to think about your mode of action. Do you need to have an enzyme inhibitor? Do you just want something that binds to your protein so you can stick a warhead on the other end to get it degraded or get it dephosphorylated to inactivate it? What's the mode of action you're after? Are you trying to compete with a nanomolar peptide that's in the cell, or are you trying to compete with a micromolar biologic? Is that micromolar biologic unfortunately knocking around in the cell at millimolar concentrations, in which case you're never going to compete the damn thing, so you might as well give up now and do something else, KRAS. So consider your mode of action. But even more than that, for all of these kind of hit ID methods, the whole of the drug discovery process is a very, very, very dodgy pile of stones. This is the best analogy, that has a habit of falling over at the slightest excuse. So you try and be as confident about each level as possible, especially when you get quite a long way down. Most drug targets turn out to be no good because the target itself is not a viable biological target. So that's where most campaigns fall over, because when you do finally make a compound with cellular activity, it doesn't do anything in an organism. Or there turns out to be redundancy pathways, or the cells mutate their way around it, or something really irritating. So the basic one is you'll need really well validated target. But once you've got that, the next point at which everyone gets horribly mistaken and misled on is that interaction between the ligand and the target. So just because it inhibits an assay does not mean it's a mode of action that you want. It does not mean it's a mode of action that could be turned into a drug, and I'll come back to that in a minute. But everything rests on this kind of hit ID method. Well, everything initially rests on the fact that is the target any good. But assuming the target's any good, then your chemical matter has got to be robust. So in terms of thinking about the steps, is the ligand what you think it is? That's not a joke. The number of compounds that you buy from people that are not real is horrific. I'm a chemist, analytical chemist originally, so about a quarter of the things when you buy them from vendors aren't what they claim. So do check that if you've got an active thing. Is it stable? So I don't have it in this deck. It's a different deck when I go on about assays for hours. But basically, we have examples of compounds that have degraded in DMSO or in aqueous and have gone from being active to inactive, or gone from inactive to active. It can happen in any direction. Okay, so that time, if you're seeing inconsistency between assays, you need to start thinking about, is the compound degrading in some way? But it could also be degrading into an active species, which we've had, which was hilarious. Is the protein what you think it is, or is the target, if it's not a protein, is it what it think it is? Is it folded correctly? Is it folded relevantly? Does it have any necessary post-translational modifications? Should it be glycosylated? When you get to the bottom of the list, by then you're saying, do they actually interact? So, is your assay actually identifying an interaction, or is it just identifying an artifact onto one of the other points further up? And then finally, if you can get it, what's the structural basis for that interaction? Because that, although you don't need a crystal structure, it really makes life easier if you can understand the structure of what's going on. The reason for doing that is because what we don't want is the tire to come off, which is the best photo ever. The number of projects where halfway into it, 2 years down the line with 10 chemists working on it, you discover that the original hit matter was wrong is really quite noticeable. And again, I'll come to that in a second. So thinking of the small molecules and having a curation on your screening library. Two examples here: bosutinib, that kinase inhibitor at the top, clinically approved. For 6 years, the thing labeled bosutinib isomer was what you bought. If you bought bosutinib from a vendor, you bought bosutinib isomer because someone had made it wrong, had done the analytical chemistry wrong because they look very similar, and what they were selling was the isomer. For 6 years, there was only one company made it. It was only when someone did a high-res crystal and noticed that the density was completely wrong that they actually found that out. So for 6 years, that was the wrong compound, and that's a clinically approved compound. That TIC10, TIC10 from 2 patents, that's now finally been sorted out after nearly 10 years of legal wrangling, because they found a compound that looked quite promising as an oncology. Do you know this story? This is lovely, this story. It looked like it was a very good oncology target, had a good effect. They patented it. Unfortunately, they patented the wrong structure because they did the analytical chemistry wrong. Someone else bought the compound, noticed it wasn't the one that had been patented, and then patented the correct structure for the same compound. And it then took the lawyers nearly 10 years and an awful lot of dollars to sort that one out. It's horrific. And it's just because they hadn't actually worked out what the compound was. So make sure you've got the correct compound. Impurities, metals screw up so many assays, especially biochemical assays, and they're really hard to detect. And there's loads of publications. There was a beautiful one from Dundee a couple of years back where they had a hit which looked absolutely perfect in all of the biophysical methods until they finally got a high-res crystal, and it turned out to be zinc. That was a zinc contaminant. So try making your compound. So if you think you might have a metal contaminant, make the compound in a different way. Buy it in a fresh stock. Try and sort out some sort of separate way of getting at it. Low levels of potent impurities. If you can get cross contamination in a lab as well, that happens in synthetic chemistry. You can, if you're working on a lead series and you've got a backup series as well, you can end up with contamination in the backup series. This is one of the examples from the paper, which we'll come on to in a second. Compound stability, I've touched on. Aggregators, this is great. So everyone knows compounds aren't, small molecules often have solubility problems. You can see it looks like milk if it goes revolting. But even below that level, you can have hydrophobic particulates and aggregates floating around in solution, which sequester protein really nicely. So protein will come along, nice hydrophobic ball of grease in suspension, protein sticks to it. You can't see anything, but your protein's now unfolded. Most biochemical assays, if you take the protein out of the system, that will register as inhibited. If the protein is now not there because it's stuck to the particulate, that looks like inhibition. So many compounds, the mode of action of the inhibition is not in a useful way, binding to the protein, but just forming a glob of grease and taking the protein out of solution. And we had an example where, which was discussed at one of the FBLD meetings, where someone, when they transferred an assay between 2 labs in the same pharmaceutical company, the decimal point on the detergent had shifted by 1. Simple as that. And what was originally a hit then got optimized over the next year with a team of 20 people working on it into aggregators, because the assay was reading out as positive if it took the protein out of solution, and they just optimized the aggregation properties of the compounds. Worse than that is this one, which is where this was looking for amyloid, inhibition of amyloid fibril formation, and hydrophobic particulates do it beautifully, but by forming particulates. So the fibrils then stick to the hydrophobic aggregates. So you are inhibiting the action, fibril formation in this case, but not in a way that you want to do, because that's never going to be a therapeutic. Okay. I'm sure people know about this, but I'm going to touch it anyway. Pains. So pan-assay interfering compounds. So these are these guys. So this was the cartoon that was done in Nature to try and describe them. But there are a whole load of compounds that keep turning up over and over and over again in the literature as people identifying them as active in biochemical assays or active in different assay formats. And they're things like redox reagents or metal chelators or polyphenols, covalent modifiers, all manner of just horrible modes of action that are never going to be any useful as a therapeutic, but will ring positive in an assay. So they try to come up with these pains filters to take out some of these motifs, but there's always new things that can go wrong. So you kind of need to be aware of that. And this is this other paper that I was mentioning. So I published this back in 2013, which was nominally focused on fragment screening, but was really looking at assay artifacts in early hit ID. In particular, things like in crystallography, people solving the density, and it was actually PEG in the binding site. The story about the optimizing the compound for aggregate formation came from this paper. We set up an anonymous Gmail address, and people were sending in horror stories that they wanted to get off their chests from different projects. So we had this wall of things that where where projects had gone wrong, and they just kind of wanted people to learn of it. It happens so often, and it's very hard to publish. It's very hard to make people aware of it. But being aware of assay artifacts before things progress into chemistry and making sure it's really robust is absolutely vital. So I'm going to quickly do this one. Not going to get all these slides. I'm going to quickly do this one because it's quite fun. You may have seen this floating around LinkedIn, but I think this is a beautiful example of understanding your assay and survivorship bias. So in World War II, the American Navy did a survey trying to understand where the damage was done to aircraft because they wanted to put armor on the aircraft. And armor's very heavy, so those planes down, they can carry less. So you don't want to put armor everywhere. You've got to armor the bits that take damage. So they did an analysis of all the planes and said, okay, the big red spots is where the damage gets taken. Let's armor these. That makes complete sense. This statistician, Abraham Ward, who was an Austrian refugee, which is a whole different get of the fish story because they didn't believe him for years because they thought it was a German spy, pointed out that their survey only looked at the aircraft that have made it back after flying a mission. So what you're actually assaying were aircraft that were not shot down. So the readout of their assay, their survey, was where can a plane take damage but still get back, not where does a plane take damage and crash, which was the relevant bit. So Abraham Ward said, armor the bits where there's no damage taken because those are the vulnerable spots, assuming damage is taken evenly, which you had to prove scientifically. So the point of a slightly rambling anecdote is consider what your assay is actually telling you, not what you think it's telling you. Take a step back and say, what artifacts could be reading out in my assay as positives, not the one that I necessarily want. For example, if my protein is being taken out of solution by an aggregate, would that show up as a positive? Yeah, and what assumptions are you making? And then the other, that's the book that came from originally. So the other brilliant thing is to look at the data. So this is a statistical data set which has the same mean and standard deviations and correlation functions, and every now and then there's something in it which is interesting. But if you just look at the raw output, you miss it because the numbers don't change. I think this is the most beautiful example of data manipulation, because every now and then there's a dinosaur. But if you just looked at the raw text output of the assay instead of trying to dive down into what the actual pattern was, you miss the dinosaurs, you miss the circles. Okay, so going to focus a bit more on the actual chemistry and the lead identification and lead optimization. Skip over this one because it's not that interesting. HSP90. So HSP90, well-known chaperone, cancer target. This is a hit that came. This one actually came from a virtual screening campaign run on HSP90. Sub-micromolar hit, so Ki, inhibition constant of a couple of hundred nanomolar. That's the crystal structure of it. And what you have, oh no, don't touch anything. What you have is a network of hydrogen bonds. So you can see this network of hydrogen bonds between the compound and the protein that's holding everything in place, including that there's an absolutely conserved aspartate up the top. But as well as the interactions that the compound is making, you can see how you could optimize it. So for instance, the methyl on that pyrazole coming off is a potentially good potency handle. There's pyrrole, there's places to go there. The bicycle that's coming out of the plane towards us is a nice conformational constraint, but could be optimized further. the pyrrole there can be modified to change the strength of those hydrogen bonds, for instance. So you can see that you've got the basic interaction there, the basic chemical matter is correct, and we're sure of it because it's been through a whole load of orthogonal assays. It's active in lots of different systems. We've got a crystal structure of it, but now we need to optimize it. Okay, so and there's space to do it. So so that's kind of our lead that we've identified that we're going to optimize. And the things that we're now trying to optimize is not just potency, but it's things like the absorption, the way, so how rapidly does it actually get into an organism? How does it distribute across the organism? Does it all pile up in the wrong place? Does it get metabolized? Does it go straight back out again? Does it need to be IV? Is it oral? So there's all these other parameters as well as affinity, as well as efficacy in whatever system you're looking at, which you have to optimize. And that's that progression from a ligand or an inhibitor through to an actual drug. And we need to balance all of these while still maintaining the function of the first bit. That's just a list of things that are measured. Drug-drug interactions are horrific, and metabolism is horrific. But anyway, so kind of glossing over that for time. You need to make sure that although the compound may still be active in a biochemical assay, is it active in the cell? So this is kind of a cell-based readout. We know that it's active in biophysics. We know it's active in NMR. We know the crystal structure. We know the ITC data. So we know the calorimetry. We know the SPR. This is a compound which is supposed to inhibit HSP90, so you would expect it to decrease the client proteins. So HSP90 is a chaperone that helps proteins fold. If you inhibit it, those proteins should not fold correctly and should be degraded in the cell. If you now do a Western, if you're looking at a client protein, as you titrate the compound up, you see a drop in the client protein because it's not folding properly and it's being degraded by the cell. Okay, so that's kind of an example of target engagement, or at least you're hitting the correct activity and the function in the cell from a Western readout in that case. So that was the lead series that we mentioned as identified a lead. But it's got a pile of issues. So it's not novel when we found it. So it was covered by someone else's patent. So that's no good. Potency wasn't that good. It's not that selective, so it inhibits GSK3 beta. It's not particularly soluble. PK is dreadful, and it's an inhibitor of P450. So you get drug-drug interaction. So it's not a drug. It's got a whole load of things we need to improve. So understand, and I apologize for this ancient picture. So ancient cartoon, but it's showing the idea that you can grow it. You understand the structures. You can guide particular regions of the compound that aren't interacting with the protein to try and optimize those other parameters. So for instance, by growing out, we said that selectivity handle, not the selectivity handle, the potency handle, that methyl, then we can build an amide. I just have to point at it. We can build an amide on there that then makes interactions with G97 and lysine 58. So we're picking up a whole load of extra hydrogen bonds with the protein. So we're picking up extra affinity. We're making extra interactions. So this kind of increases the potency whilst not doing anything actually bad. Another one was changing the pyrazole to an isoxazole, which gives a stronger hydrogen bond. So, and you can see that, so although in the crystal, it's got the same hydrogen bonding pattern, which is down at the bottom, and here, this is the matched pair. So again, with kind of that kind of lead optimization, you do a lot of matched pair analysis, trying to make a very small change to the compound. In this case, isoxazole to pyrazole on that 5-membered ring, just introducing that oxygen. The hydrogen bonding pattern is the same in the crystal, but if you look at the kinetics in the SPR, it's now slower on and slower on and much slower off for that pyrazole. So you're making a stronger hydrogen bond. It may need a bit of rearrangement of the water structure, hence the slight drop in the on rate, but the off rate is really reduced. So once you're on, you stay on, and that's the state that we want. So you can see the optimization of the affinity parameters, but then also coming out, I mentioned that bicycle as being the kind of the conformational constraint. You can keep that first ring, but then grow out into a morpholine to improve the solubility and improve the way that the compound is treated, behaves and distributes in the body. And you can also modify the metabolism of it by changing that ethyl to isopropyl. So again, that sterically blocks some of the modification and the metabolism by the body. And it's those kind of little tweaks. So the difference between that first compound and the second, and the one on the left and one on the right, isn't huge, but that's now gone from a lead to potentially a drug. So that's now looking like a preclinical candidate that we can take into clinical trials. And it's that kind of solid basis of understanding how it's interacting, and then understanding how you can then tinker with that to change the other properties to optimize it into being a drug without breaking the interactions and so on. That's kind of the really important bit. So in animal models, so you then put that compound into animal models and see what happens. And hopefully the animal model is a good reflection of the human, because that's what we want to be treating, but you've got to start somewhere. Obviously starting with cell culture is absolutely the first place to start. So this is a xenograft on a mouse, and you can see the NVP AUI922 was the preclinical candidate, having a really nice effect on the xenograft size and the biomarkers, and then also having not good in vivo efficacy again in that animal model, and it's still in the things across it. It's in just over 20 clinical trials still. Seeing if it's any good. It probably won't be, because the target itself has liabilities. So HSP90 has some inherent liabilities as the target, it turns out. But the compound does what you want it to. Okay, so, and then just something I wanted to touch on. Oh, it's back on time. In terms of clinical trials and formulation, because I haven't really discussed formulation at all, but actually how you present that therapeutic agent to the body. Remdesivir is an antiviral. It's active against COVID-19, SARS-CoV-2, and it's a really nice inhibitor of the viral RNA polymerases. A very good antiviral compound, but it's horrific in terms of solubility. It's a dog. It just goes straight to the bottom. You can't give it to people. But what you can do is to formulate it with a cyclodextran, which then keeps it in solution and gets it to the site of action. So that's the cyclodextran called Captazole, which is basically like an open bottom bucket, and the remdesivir sits in the middle, gets transported to the correct part in the body, and then dissociates from the cyclodextran and can be active. And it's at such a low concentration by the time it gets there that the low solubility isn't an issue. It's only really an issue when it first hits aqueous solution. So you can kind of modulate that with the formulation. Formulation is a black magic as far as I'm concerned. It's amazing and very complicated, and you can do clever things. But anyway, so that's kind of what you can do with formulation. And then the final bit of the whole drug discovery, once it's been through phase 1, phase 2, phase 3, which I'm not going to discuss because that would be a whole different thing. And it's been approved. It's post-approval and keeping an eye on what the drug does, and that comes looping back to thalidomide and induced proximity. So thalidomide, everyone knows it was only marketed for 6 years. Substantially more than 12,000 children born with birth defects. Absolutely horrific. Everyone knows about it. It's very widely studied, and most of the current regulation is based on thalidomide and the way it happened. But over the last decade or so, the biological mode of action of thalidomide has finally become understood, and it's opened up this whole induced proximity field. So there's a developmental transcription factor in humans called SAL4, which is not present in rabbits, which is what they tested it on to see if it had any effects. SAL4 is a human transcription factor. SAL4 is not recognized by cereblon, which is part of the cellular degradation machinery, unless thalidomide is there. Thalidomide binds to cereblon and changes the recognition surface at the end. So what the bit that recognizes the protein to be degraded is changed when the compound binds by thalidomide binding. That creates, it's got a neo-substrate. So SAL4 is now degraded by cereblon. So it's the cellular machinery is degrading the wrong protein because the recognition surface has been modulated by the binding of cereblon. So what that means is, the net upshot is, if you are unlucky enough to have thalidomide and you're pregnant, then you get SAL4 degradation, which causes a whole load of teratogen effects. If you don't have the thalidomide, cereblon does not recognize SAL4, it's not degraded, it acts normally. And so that modulation, understanding that route for the pathway of degradation was what kind of led into the induced proximity PROTAC type approaches, which is now, they're now in clinical trials for where you are recruiting and degrading proteins that you want to degrade. So for example, if you have an undruggable protein, because the protein is undruggable, because you can't inhibit it, perhaps you can degrade it instead. And that's been quite a nice example with some of the kinases and the pseudokinases, where you can degrade them by binding to the kinase domain, even though the kind of the disease effect is coming from one of the structural domains that you couldn't necessarily interfere with. So that's kind of the really interesting new mode of action. So, and then just to finish with the really kind of depressing slide. So basically it takes years. So 13 years, 8 to 13 years-ish, if you're lucky. It can go a bit faster. It can also go a hell of a lot slower. So we've had compounds sitting in phase 1 for 5 years while you try and recruit enough people. That's one of the amazing, you know, if it's a rare disease or it's a hard to measure disease, it's really hard to get the number of people. So really good compounds can just sit and languor. So anyway, 10% of programs that generate a hit reach the clinic, and roughly 10% of compounds that make it into the clinic make it into the patient. So that's why it's so horrifically expensive and goes wrong all the time. And so finding ways to not have it go wrong all the time is you know, quite interesting. Hence AI, amongst other things. So and moving away from some of the other antiquated ways of doing things. 4 o'clock on the nose. Any questions? That's me done. Any questions or thoughts or things?
So a lot of my job is supporting academic researchers who are that target discovery. I always talk, my favorite projects are a researcher spent 15 years looking at some system, some protein, and then goes, oh, this is in a disease state and I think I can interfere with it. Suddenly very exciting, right?
Yeah.
But then there's the dichotomy that to get any sort of substantial funding in, it's a race to getting some in vivo efficacy data, because people won't give you millions until you do that, because you're just a biologist playing around. So you must get a lot of people come to Vernalis and go, hey, I've got a target. What is the cheapest, fastest route for you to give me something, do in vivo efficacy? What's your like sales pitch to I guess, take your time?
So absolutely, the showing target engagement and the mode of action being what you think it is absolutely vital. That's something we've done for quite a lot of academics. So they come to us and say exactly that. I think investors are becoming a lot wiser than they were. So now, if you can show biophysical engagement so if you can show a direct interaction between your compound and the target that you think it is. So ideally a crystal structure, but SPR, NMR, whatever, to show that direct interaction between your compound and your protein. Investors really rate that. As you know, obviously you need a cellular effect as well, but you need that evidence of direct target engagement, because too many times it's been just chasing a shadow of an activity in cells and you never know what it is. And that takes back to the phenotypic screening, where if you don't know what the mode of action is, if it's not what you think it is, you end up optimizing for the wrong thing and wasting an awful lot of money. In terms of kind of alarm bells or triggers, single digit micromolar activity in cells is the worst thing that we can see ever. So many things work in single digit micromolar in cells, and then that doesn't translate to anything else. And that's because almost always because the compounds are coming out of solution in cells. So they're not that soluble. They come out of solution, they cause particulates, it sequesters something which has an effect on the cell, which looks like what you want it to look like. And that's kind of the biggest red flag for us, is a single digit micromolar in cellular activity. If you have SAR, on the other hand, so if you've got nice, well-defined SAR, where you can see a whole compound series of 20 compounds with a 10-fold range of activities from, say, 100 nanomolar to, okay, 100-fold range of 100 nanomolar to 10 micromolar, that's really valuable, because then you know that the SAR makes sense. If the SAR doesn't make sense, or there is no SAR, then it's much more likely to be an artifact. If you've got that range of SAR, then we can probably optimize it to make a cell active compound to test the target. So in terms of how much effort is it to get a cell active compound, it's not that hard. So 18 months, 12 to 18 months, not that many compounds to get a decent activity. But you need that fairly firm footing to begin with, and that's, like I said, that's what we see a lot of is artifacts coming in.
I don't know if there's any questions online or anything, but I have one other question that's more granular. So you talked about these aggregators and this being a flaw, and I guess you've touched on a little bit. So you talked about 20 chemists working for ages and then finding out this. Is there an assay you can do to check for this, or is it?
Absolutely. Yeah, totally. So measure the solubility. So ideally you want at least a 10-fold differential between your activity and your solubility. Okay, so if you've got 1 micromolar activity in the cell, you really need 10 micromolar solubility in the cell medium, and ideally quite a bit higher than that. So you want that clear differential in terms of solubility versus activity. The other thing is to plot activity against something like log D, or measured solubility, and then, or PSA or something. Just try some different things and see what correlates. Because if it's just something to do with the physchem parameters of the compound, not a direct effect on your target, it will correlate with something else. So like hydrophobicity. But you can measure solubility in lots of, so we measure lots of solubilities. NMR is another great way to measure solubility. So.
Excellent. Are there any other questions? Nothing online? Okay then.
Cool.
I think we're done. If everyone can thank Ben.